So today let us start the chapter Humoral and Cell-Mediated Immunity. It is the continuation of the chapter Immunity. In immunity class, this is the continuation of that class. We have seen that spatially immunity are of two types. That is our innate immunity, that is the inborn immunity, and acquired immunity, or the immunity which is acquired from any external sources, from anywhere, either from anywhere or from someone. Right? So, the acquired immunity is always specific in nature. It is very much specific to the specific pathogen or to the specific foreign particle. Right? So, specific immunity, that acquired immunity is specific, and the specific is actually humoral and cell mediated. They are of two types. See, specific are of two types: humoral and cell mediated immunity or cellular immunity. So what is humoral? Actually, it involves antibodies secreted from B cells or beta lymphocytes. So B cells dissolved in blood plasma. Again, I repeat, what are humoral? So there are some features of humoral and cellular. In case of humoral, it involves antibodies secreted from B cells dissolved in blood plasma, demonstrated as an immune response using only blood serum, and defense against bacteria, bacterial toxin, and viruses. This is humoral immunity. Now, cellular, it involves the activities of specific white blood cells. That is only the activity of T cells, specific T cells. Defense against cancer cells, virus infected cells, fungal, parasites, and foreign cells from transplants, from any other transplants. So they are also defensive against fungal cells, parasitic cells, and foreign cells from any kind of transplants. Right? Okay. Now we will learn some terminologies. Uh, for that, first we need to know what is CD4. It is the name of a glycoprotein. It is a name of a protein, CD4. It is a glycoprotein found on the surface of immune cells such as T helper cells, monocytes, macrophages, etc. It is found on the surface of immune cells like T helper cells, macrophages, monocytes, etc. Okay. Now B cells and T cells. You have seen here B cells and T cells. So what are those? B cells is B lymphocyte and T cells is from T lymphocyte. So a lymphocyte developed in the of bursa of birds. So it is a lymphocyte which is developed in the bursa of bats and bone marrow of other animals that produces antibodies and is responsible for immune system, even in human blood. Also, the human antibody, in the structure of antibody we have studied that B cells are present. So even in human also B cells are present. So it is a lymphocyte developed in the bursa of bats and bone marrow of other animals that produces antibodies and is responsible for immune system. Clear? And T cells, it is also a lymphocyte from thymus gland. Okay, it is secreted, it is developed, not secreted, sorry, it is developed from thymus gland. So from thymus that recognize specific antigen and can activate or deactivate other immune cells. It can either activate or can deactivate other immune cells. Right? So you have come to know what is humoral, what is cell mediated, what is CD4, what is beta cells, and what is T cells. So we'll move on to the activity, how humoral and cell mediated or cellular immunity occurs. Now this portion is humoral, and from here this is the cell mediated one. So first humoral, it takes place in two steps, 1 and 2. See, these are the bacteria or the foreign particles, you know, cocoid, spherical shape and little elongated shape. These two types of bacteria are found. Okay, so these are the foreign particles. Now, B lymphocytes are present in the body, right? B lymphocyte with antibody. Now, this B cell secretes antibodies. See, these are secreted antibodies. And you know, antibodies are Y-in shaped antibodies. With these antibodies, these foreign particles gets attached. See, antigen antibody binding is taking place. See, antigen, and here this is the antigen, and antibody is this secreted from B cells. So these foreign particles acts as antigens here, and you know that antigens binds with antibodies, so they come and binds here. See, it is binding is happening. After binding, what happens? The pathogen clumping takes place, and after that these antibodies helps in the neutralization or the lysis of the antigens or the foreign particles, and see, cell lysis occur. You can see from here, this is a fully encoded bacteria. The upper portion or the encoded portion or the cells, the outer cells are dissolved or neutralized through cell lysis. Okay, so as cell lysis occurs, and this process is called phagocytosis process. So as cell lysis occurs, they become inactivate. Okay, they are no more stronger to develop a disease. They become less means they become less strengthful. Okay, they are not strengthened enough to develop a disease. So cell lysis takes place. So after pathogen clumping, cell lysis takes place. After this binding, this antigen antibody binding, antibodies neutralizes the antigens or the foreign particles, and cell lysis of the take place. And this process is called phagocytosis. And see, phagocytosis means engulfing the pathogen. So a cell is engulfing the foreign particle and neutralizing that particle. So this process is called phagocytosis, and this foreign particle acts as macrophage to that larger particle in which it enters. And this macrophage enters and this cell engulfs that macrophage. This process is called phagocytosis. Okay, now in second step what happens? See, B cells, in the first cells what happened? B cells activation requires two signals. What are the two signals? Binding of beta receptor with antigen. Here binding with beta receptor with the antigens takes place. Beta receptor, from beta cells these antibodies are secreted. That means from these beta receptors or B receptors the antigen binding takes place, and then phagocytization or cell lysis occurs. That is the killing of pathogen through the process phagocytosis. Okay, this is the very easy form I have done to you to remember. This is a very easy form. Okay, and the second signal is interaction of B cells with T helper cells. This we'll study here. See, second step. Now this B cells releases CD4. You have seen what is CD4? It is a glycoprotein found in the surface of this. T helper cells, monocytes, macrophages. So now this B cells secretes or starts releasing CD4, and T helper cell is there which help in immunogenic process. Helper means it helps the others. It helps the process. So now T helper cells join with beta cells. Okay, and from there what happens? Two things or two cells are produced. After their reaction, two cells are produced. What are those two cells? Memory B cells and plasma cells. Memory, memory, once you catch hold of memory, you will never forget. See, your memory also works like that. Our human memory also works like that. Once I've seen this thing or this house or this person, we will never forget when we see them or those things in the second time, because our memory stores that image. Okay, and we can flashback that where we have seen this person or where we have seen this thing or where or what was this thing, we can remember because our memory stores that image. Our memory stores that signal. So this B cells also act as memory B cells. Now these B cells can now recognize the foreign particles. They gain the memory. That means this memory B cells act as the memory here and can identify the particular pathogen or the particular foreign particle and will neutralize that as it have catch hold of its memory, and next time that particular disease will not occur, because as soon as this pathogen will enter, once the memory B cells have produced in the body against that particular pathogen or bacteria or foreign particle, whatever you say, these memory B cells can recognize them if they try to enter the second time, and they that pathogen will unable to create or unable to uh, focus on any other diseases to occur in our body. So this memory B cells can recognize them, can identify them, and will neutralize their activity, right? And these are the plasma cells. These are the plasma cells from where again the antibodies get secreted. So the beta cell activation requires two signals. One is binding of beta receptor with antigen and the interaction of B cells with T helper cells. The interaction of B cells with T helper cells. Now B cells may be differentiated into memory B cells or plasma cells. And these plasma cells can synthesize millions of antibodies specific to an antigen. See? Now these plasma cells, these are the plasma cells, and it can synthesize millions of antibodies specific to an antigen. So these B cells split up and helps each other. Memory cells, as I said just now, that they can recognize and can identify the pathogen. Now the antibodies specific to that antigen or to that pathogen is secreted by the plasma cells, and that antigen and antibody binding again happens, and in the same process, after this antibodies binds with those specific antigens, again the neutralization occurs. So after this, again this process occurs, and that pathogen clumping and neutralization, that means through phagocytosis, and that foreign particle will be destroyed in our body. It will not able to create any disease in our body or to establish any kind of disease in our body for the next time. Clear? And cell mediated cellular, here actually T cells, that is the precocious cells, are of three types: helper T cells, cytotoxic T cells, and suppressor T cells. Now, out of those three, this helper T cells acts in humoral immunity. Here it takes place. And suppressor T cells, what does it do? It stops the phagocytosis process. If this phagocytosis process goes on and on, then what will happen? All the cells lysis will occur. So somewhere this process needs to be stopped. So the suppressor T cells are there to stop the phagocytosis process in our body. So only when the external body will enter our body, so the helper T cells will help the B cells to produce these two cells further, and antigen antibody binding will further neutralize those pathogenic cells or antigens. And somewhere this process needs to be stopped. For that, suppressor T cells helps and stops the phagocytosis process. Now T cell receptor. Now T cell receptor. They are of two types: activated helper T cell and activated cytotoxic T cells. So this activated helper T cells is used in humoral immunity, or the process in humoral. Now, in cell mediated, this activated cytotoxic T cells actually play the role. And this activation of cytotoxic T cells, when they activate, they release two types of proteins like CD4. Now they are activated, means they are charged or they are energized. So plus they are charged particles. So they are activated, means CD4 plus and CD8 plus. Two types of strong proteins are being secreted. Now what do they do? Now CD4 plus destroy the infected body cells. All the infected cells in our body are being destroyed by this protein, and CD8 plus bind to infected cells, and they bind to infected cells, bind to MHC1 protein. What is that MHC1? MHC is major histocompatibility complex. So major histocompatibility complex. Now those infected cells, this protein binds. And then further reaction takes place, and ultimately the disease establishment is stopped. So these are the two major types of specific type of immunity. And like this, the process and the actions are being taken. So this is a very easy explanation, and stepwise it is very much easy to remember how this humoral and cellular immunity takes place. Is nothing important, nothing very much confusing. So this is a very easy explanation, stepwise I have provided. I think it will be easy for you all to remember also. So for today, it's still this much.